Chapter 2 was all about how atoms come together to form molecules, the different types of bonds, things like that. Chapter 3 is, let's look at 4 special types of molecules, what they call macromolecules: carbohydrates, lipids, proteins, nucleic acids. How they're formed, what their roles are, things like that. Chapter 4, let's take those macromolecules, let's put them together. Let's put them together and make a cell. I could take pretty much any living thing, as long as it's multicellular, multi-tissue, multi-organ organism. I could take any animal, plant, whatever, take a scalpel, carve a chunk out of it. So with an animal, I can keep that organ alive for a few days in my lab. I take that organ and the scalpel, slice and dice some more, cut that organ down into tissues. Well, I can keep the tissues going a lot longer than I can the organ. Instead of a few days, maybe a week or so. I take the scalpel and I cut up that tissue into individual cells. Well, I can keep those cells going for a long time. But the moment I take a scalpel and I cut open a cell, I have nothing. I have a soup of phospholipids, proteins, nucleic acids. Because of that, we now think of the cell as the building block of all living organisms. It could be one cell, it could be multi-cells. Cells as the unit. Get a bunch of cells together, working together, you get a tissue. Get a bunch of tissues together, you get an organ. Get a bunch of organs together, organ system. Enough organ systems, you have an organism. But it's all built on a cell. Now, cells generally are too small to see. Okay? So you need some form of a microscope. The microscope you see there on the left, that's pretty much quite similar. It's an old one, but it's quite similar to what you would see in the classroom lab. That big honking thing you see on the right, well, that's a transmitting electron microscope. The one on the left, this one, uses light. This one uses lasers. What limits us, we can't see single cells. We can't see single cellular organisms, so we need some sort of microscope. Cheap microscopes, magnifying glasses, may magnify 3-4 times, but you still can't see stuff. They look like specks. Cheaper microscopes can magnify, but everything gets fuzzy. You want nice, crisp, clear pictures like the one you see there. Well, you need a microscope with really good magnification and excellent resolution. Now, the rest of this lecture here is going to be talking about microscopes, how things work. This isn't test material. This is to give you an appreciation. So when you see pictures in textbooks, so you have an appreciation of what it took for that person, that scientist, to take that picture. Now, magnification, no big deal. You magnify something, you make it look bigger to your eye. You've done this as little kids with, magnifying glasses. So it's just enlarging, making look bigger. But the more you magnify, the more resolution becomes a problem. You look on the left, good magnification for this single cell, what they refer to as a radiolarin. The single cell makes a coating, a protective coating, a cuticle around it. The problem is the light microscope uses literally a light bulb, shines light through. It's hard to see stuff. Everything's fuzzy. Magnification high, resolution low. But you go over here to this electron microscope. Doesn't use a light bulb, it uses a laser. Single electrons bouncing off. Computer calculating what that means, the way the things bounce off, reflect, refract. High magnification, very high resolution. You can see the structures that are only hinted at here under a light microscope. Now with light microscopes, that electron microscope, those are going to be $500,000 to a couple $1,000,000 each. Yeah, they're not going to get one of those. Light microscopes, not too bad. Get what's referred to as a compound microscope. It sends light through and around what you're trying to look at. You see here on the left this cell. You put special filters, you bend the light in specific ways, you now get to see, instead of this ghost of an image, you now get to see the shadow. Allows you to see some of the intercellular organelles. But again, high magnification, low resolution. That's why we go with the electron microscopes. You have what's referred to as transmission. You bounce a laser through the cell. Whatever shadows comes up black. So you see here a mitochondria. You see the membranes, the cristae inside the mitochondria. You see around it vacuoles, vesicles. So instead of just seeing the cell, you see in high resolution organelles within the cell. Whereas scanning, which is right here, you bounce the laser off the surface of the cell. You see what it looks like in 3 dimensions. Transmission goes through, scanning bounces off. It wasn't that long ago, actually, just about 150, 200 years ago, where we came up with microscopes, rudimentary that they were, and we started to develop what we call cell theory. Cells are the basic unit of life. All living organisms are made of these cells. All cells come from existing cells. You go back a couple hundred years, they were under the impression of spontaneous generation. Cells just poof, were there. Sometimes they look like pre-existing cells, sometimes not. We do know that all living cells have 4 things in common, whether you're talking prokaryotes or eukaryote. There's the plasma membrane, the cell membrane. It's basically a boundary. Inside, outside, outside, inside. Keep things in, keep things out. It's a lipid bilayer, so those phospholipids. All living things, all cells have this. Now, depending upon which cell types you're talking about in which organisms, different types of phospholipids will be present in different ratios. There is a cytoplasm. Cytoplasm, which is made of cytosol, which is an aqueous solution. Aqua, water. It's a water-based solution that has lots of sugars and salts and amino acids and all sorts of things. It gives it more of a gel-like consistency. It aids in things being able to float. There's going to be some form of DNA there. One or more chromosomes. And then there's going to be ribosomes. Remember ribosomes from last chapter. Ribosomes read the messenger RNA and make the primary structure of a polypeptide.